Welcome. Today, we're diving into the fascinating world of genomic editing research. Whether you're a student, a scientist, or simply curious, understanding how to get started in this field is essential. Genomic editing allows us to make precise changes to DNA, opening doors to new treatments, agricultural advances, and more. In this video, we'll guide you step by step from understanding the basics to planning your first experiment. Let's begin by exploring what genomic editing really means and why it's such a groundbreaking area of research. Genomic editing refers to a set of technologies that enable scientists to modify an organism's DNA. The most well-known tool is CRISPR, but there are others like TALENs and ZFNs. These tools can add, remove, or alter genetic material at particular locations in the genome. The ability to edit genes has revolutionized biology and medicine, making it possible to study gene function, model diseases, and even develop gene therapies. Understanding these technologies is the first step in starting your own research journey. Impact of genomic editing is enormous. It's transforming medicine by enabling targeted therapies for genetic diseases. In agriculture, it's helping to create crops that are more resistant to pests and environmental changes. By studying genomic editing, you're positioning yourself at the forefront of scientific innovation. This field offers opportunities to solve real-world problems and contribute to advances that can benefit society as a whole. Before you dive into genomic editing research, it's important to have a solid foundation in molecular biology and genetics. Understanding how DNA, RNA, and proteins interact will help you grasp the mechanisms behind editing technologies. If you're new to these topics, consider taking introductory courses or reading textbooks on genetics and cell biology. This background will make it much easier to understand the protocols and results you'll encounter in genomic editing research. Next step is to get to know the main genomic editing tools. CRISPR-Cas9 is the most popular due to its simplicity and efficiency, but TALENs and ZFNs are also widely used. Each tool has its own strengths and limitations. Spend time reading scientific literature and watching tutorials to understand how these systems work. Knowing the differences will help you choose the right tool for your research goal. Crucial part of starting any research is reading the foundational papers in the field. Look for review articles and recent studies on genomic editing. These will provide an overview of current techniques, challenges, and breakthroughs. Reading widely will also help you identify gaps in knowledge and potential research questions. Make it a habit to stay updated with the latest publications as the field evolves rapidly. Connecting with others who share your interest in genomic editing can be incredibly valuable. Online forums, social media groups, and professional networks are great places to ask questions, share resources, and discuss new findings. Engaging with a community will keep you motivated and help you learn from others' experiences. Don't hesitate to participate in discussions and seek advice from more experienced researchers. Having a mentor can make a huge difference when starting out in genomic editing research. A mentor can guide you through the learning process, help you avoid common mistakes, and provide valuable feedback on your ideas. Look for professors, researchers, or professionals who have experience in the field. Reach out politely and express your interest in learning from them. Building this relationship can open doors to research opportunities and collaboration. Genomic editing is a broad field, so it's important to narrow down your research interests. Do you want to work on human health, agriculture, or basic science? Identifying a specific area will help you focus your reading and experiments. Think about the problems you're passionate about solving and how genomic editing could be applied. This clarity will guide your next steps and make your research more impactful. Ethics play a crucial role in genomic editing research. Editing genes, especially in humans, raises important questions about safety, consent, and long-term effects. Familiarize yourself with the ethical guidelines and regulations in your country and institution. Responsible research means considering the broader impact of your work and ensuring you follow best practices at every step. Hands-on experience is essential for mastering genomic editing techniques. If you're in school or university, look for opportunities to join a lab that works on genetic engineering. Volunteering or applying for internships can give you practical skills and expose you to real-world research environments. Don't be afraid to start with basic tasks. Every bit of experience counts and builds your confidence. Safety is paramount in any laboratory setting. Before you begin experiments, make sure you understand the safety protocols for handling chemicals, biological samples, and equipment. Many institutions offer safety training courses. Following these guidelines protects you and your colleagues and ensures your research is conducted responsibly. Before you can perform genomic editing, you'll need to master basic laboratory techniques. This includes pipetting, preparing solutions, running gels, and culturing cells. These skills are the foundation of any molecular biology experiment. Practice regularly and don't hesitate to ask for help if you're unsure about a procedure. A well-designed experiment is key to successful research. Learn how to set up controls, replicate experiments, and analyze your data. Good experimental design helps you draw reliable conclusions and avoid common pitfalls. Take time to plan your experiments carefully, considering all variables and potential sources of error. Model organisms like bacteria, yeast, fruit flies, or mice are commonly used in genomic editing research. Each organism has unique advantages and is suited to different types of studies. Consider your research question and resources when selecting a model. Learning about the biology of your chosen organism will help you design better experiments. For CRISPR-based editing, designing an effective guide RNA is crucial. The guide RNA directs the editing tool to the correct spot in the genome. Use online tools and databases to help you design guide RNAs that are specific and efficient. Double-check your designs to minimize off-target effects and maximize your chances of success. Once your experiment is planned, you'll need to order the necessary reagents and materials. This might include enzymes, plasmids, cell lines, and chemicals. Make a detailed list and check with your lab or supervisor to ensure you have everything you need. Proper preparation saves time and helps your experiments run smoothly. A clean and organized workspace is essential for successful experiments. Before you begin, make sure your bench is tidy and all equipment is functioning properly. Label your samples clearly and keep detailed notes in your lab notebook. Good organization prevents mistakes and makes it easier to troubleshoot if something goes wrong. Now it's time to put your plan into action. Follow your protocol step by step, paying close attention to details. Take your time and don't rush. Precision is key in genomic editing. Record your observations and any unexpected results. If you encounter problems, consult your mentor or colleagues for advice. After completing your experiment, it's time to analyze the data. Use appropriate software and statistical methods to interpret your results. Look for evidence that your editing was successful and check for any off-target effects. Careful analysis will help you draw meaningful conclusions and plan your next steps. Not every experiment goes as planned. If you run into issues, don't get discouraged. Troubleshooting is a normal part of research. Review your protocol, check your reagents, and consult with others to identify possible causes. Keeping detailed notes will help you pinpoint where things might have gone wrong and how to fix them. Repetition is key to reliable results. Repeat your experiments to confirm your findings and refine your techniques. Each round of experimentation teaches you something new and helps you improve. Don't be afraid to make adjustments based on what you learn. Persistence is a hallmark of successful researchers. Good documentation is essential in research. Keep detailed records of your procedures, observations, and results in your lab notebook. This not only helps you track your progress but also ensures that others can reproduce your work. Clear documentation is a key part of scientific integrity. Once you have results, it's important to share them with the scientific community. Present your work at conferences, write research papers, or post on preprint servers. Sharing your findings helps advance the field and allows others to build on your work. Be open to feedback and collaboration. Genomic editing is a rapidly evolving field. Make it a habit to read new publications, attend seminars, and follow leading researchers. Staying updated ensures you're aware of the latest techniques and discoveries, which can inspire your own research and keep you competitive. Building a network of peers is invaluable in research. Collaborate with others, join professional organizations, and participate in workshops. Networking can lead to new ideas, partnerships, and opportunities for growth. Don't hesitate to reach out and connect with fellow researchers. Research often requires funding for materials, equipment, and travel. Look for grants and scholarships from universities, government agencies, and private foundations. Writing a strong proposal is a skill worth developing. Funding can make a big difference in the scope and impact of your research. Conferences are a great way to learn about the latest developments, meet experts, and present your work. They offer opportunities to ask questions, get feedback, and find collaborators. Make the most of these events by preparing your presentations and networking actively. Genomic editing research often overlaps with fields like bioinformatics, chemistry, and engineering. Don't hesitate to learn new skills or collaborate with experts from other disciplines. Interdisciplinary approaches can lead to innovative solutions and broaden your perspective. Bioinformatics is increasingly important in genomic editing. Learn to use software for analyzing DNA sequences, predicting off-target effects, and visualizing data. Many tools are freely available online. Developing these skills will make your research more efficient and insightful. Intellectual property, IP, is an important aspect of genomic editing research, especially if you develop new methods or applications. Learn about patents, copyrights, and licensing. Protecting your ideas ensures you receive credit and can lead to commercial opportunities. Working with genetic data requires careful attention to privacy and confidentiality. Make sure you follow all relevant laws and institutional guidelines when handling sensitive information. Respecting data privacy is not only ethical but also builds trust with research participants and collaborators. Being able to communicate your research clearly is just as important as conducting experiments. Practice writing, presenting, and explaining your work to both scientific and general audiences. Good communication skills help you share your findings, secure funding, and inspire others. Collaboration is at the heart of modern science. Work with others to share ideas, divide tasks, and solve complex problems. Collaborative projects can lead to more robust results and open up new research directions. Be open, respectful, and willing to learn from your teammates. Research can be demanding, so it's important to maintain a healthy work-life balance. Take breaks, pursue hobbies, and spend time with friends and family. A balanced lifestyle helps you stay motivated and prevents burnout, making you a more effective researcher in the long run. Constructive feedback is essential for growth. Share your work with mentors, peers, and collaborators and be open to their suggestions. Feedback can help you identify weaknesses, refine your approach, and improve your research outcomes. Curiosity drives scientific discovery. Always ask questions, explore new ideas, and challenge assumptions. Staying curious will keep you engaged and help you uncover new opportunities in genomic editing research. Research is full of unexpected challenges. Be flexible and willing to adapt your plans as needed. Learning to overcome obstacles will make you a stronger and more resilient researcher. Take time to celebrate your achievements, no matter how small. Completing an experiment, publishing a paper, or learning a new skill are all milestones worth recognizing. Celebrating progress keeps you motivated and reminds you of how far you've come. Periodically reflect on your research journey. Consider what you've learned, the challenges you've overcome, and the skills you've developed. Reflection helps you set new goals and appreciate your growth as a researcher. As you gain experience, start thinking about your long-term goals. Do you want to pursue a career in academia, industry, or entrepreneurship? Planning ahead will help you make strategic decisions about your research and professional development. Genomic editing has the potential to address major societal challenges, from curing diseases to improving food security. Think about how your research can make a positive impact. Strive to conduct your work ethically and with a sense of responsibility to the broader community. As you gain experience, consider mentoring newcomers to the field. Sharing your knowledge and supporting others helps build a strong scientific community. Mentoring is also a rewarding way to reinforce your own understanding and leadership skills. Science is a journey filled with both challenges and rewards. Stay inspired by reading about breakthroughs, connecting with passionate researchers, and remembering why you started. Inspiration fuels perseverance and creativity in your research. You've now learned the essential steps to get started with genomic editing research. From building your knowledge base to conducting experiments and sharing your findings, each stage is important. Remember, research is a continuous process of learning and growth. Stay curious, keep exploring, and you'll find your place in this exciting field. Good luck on your genomic editing journey.